I hope you are well by the grace of Allah. Today we will discuss about chromosome from chapter 1. Today we will learn the physical structure of chromosome, the chemical structure of chromosome, importance of chromosome in cell division and classification of chromosome. Definition of chromosome. The thread-like structure found in the nucleus composed of nucleoprotein bears the hereditary characters during cell division is called chromosome. The term chromosome comes from two Greek words, chroma means color and soma means body. So chromosome means colored body. Discovery. First of all, in 1842, Carl Nagle observed chromosomes in the nuclei of plant cell. In 1875, A. Strasburger discovered first time the chromosome in the nucleus during cell division. In 1879, Walter Fleming named it this thread-like stainable material as chromatin. In 1888, W. Wilder finally named this thread-like body is chromosome. In 1902, Boveri pointed that chromosome is the bearer of hereditary character. These discoveries are important in your objective. Now we start to draw a chromosome. Physical structure of chromosome. This is the secondary construction. This is the primary. Okay. Now we make some thread-like structure inside the chromosome. They are chromonema. Now we double it. Now we make some dots inside it. Those are matrix. Okay, now we label it. This is telomere. They are chromonema. This is the centromere. This is the secondary construction. And both sides of centromere, there are two arms. This is one arm. This is an arm. And this is another arm. This is the satellite. This is pellicle. And this is matrix. So I have to mention the diagram name. And this figure's name is chromosome. Description of this structure, chromatid. First one, chromatid. During cell division, the nuclear reticulum breaks into a number of ribbon or thread-like bodies, which are known as chromosome. In mitotic metaphase stage, the chromosome is split into two longitudinal halves. Each of these longitudinal halves is called chromatid. Okay, number 2, chromonema. The fibrillar structure longitudinally present within the chromatid is known as the chromatid fiber or chromonema. It consists of DNA molecule. Number 3, chromomere. In leptotin substage of mitosis, some beaded structure appear to be arranged linearly along the chromosome, which are called chromomere. Centromere, the point of attachment of the two chromatids, is known as the centromere. It is known as primary constriction. When stained with basic dye, the centromere remains colorless. The centromere regulates chromosomal movement. Traction fibers become attached to the centromere of the chromosome during cell division. The centromere contains a complex system of fibers called kinetochore. Each centromere has two kinetochores linked with chromosomal arms. The kinetochore is made up of protein fibers and microtubules, which assist in the formation of spindles during mitosis and meiosis. Arm. The part of the chromosome from centromere to the end is known as arm. Generally, a chromosome has two arms. According to the position of the centromere, two arms of the chromosome may be equal or unequal. Secondary constriction. Besides centromere, one or more constricted colorless parts are found in some chromosomes. This part is known as the secondary constriction. In some cases, secondary constriction take part in organized nucleolus. Then it is called nuclear organizer. Then satellite, the part of the arm of chromosome from secondary constriction to the end where nucleolus attached is known as satellite. The chromosome that contains the satellite is called sat chromosome. The terminal region on either side of chromosome are called telomere. Telomere has a polarity which prevents other segments from joining with it. Pellicle. It is an outer covering of chromosome which is known as pellicle. Then matrix. Jelly-like semi-liquid substances inside the outer covering is called matrix. It is enriched of protein and RNA. In dividing stage, matrix become dissolved. Okay, now chemical structure of chromosome. Chemically, chromosomes are mainly composed of nucleic acids and protein. There are less amount of lipid, enzyme, calcium and magnesium ions. There are two types of nucleic acids. There is DNA and RNA. DNA is the permanent component of chromosome. Among all components, DNA contributes 45%. Actually, this is a double-stranded polymer of nucleotide molecules. Each nucleotide again consists of nitrogen bases, that is adenine, guanine, cytosine and thymine, and deoxyribose, pentose sugar and an inorganic phosphate. Then RNA. It is a temporary component of eukaryotic chromosome but permanent genetic component of some virus. Among all components, RNA contributes 0.2 to 10%. Actually, this is a single strand polymers of nucleotide molecules. Each nucleotide again consists of nitrogen bases, a ribose, pentose sugar, and an inorganic phosphate molecule. Nitrogen bases are adenine, guanine, cytosine, and uracil. Not thymine, uracil. Then protein. There are two types of protein, histone and non-histone protein. Histone protein, they are mostly basic in nature and have significant role in controlling the function of chromosomal DNA. The histone proteins are arginine, histidine, and lysine. The percentage of histone present in the protein is 55%. Non-histone proteins are mostly acidic and some have enzymatic functions. Proteins are dissolved out during cell division. Then types of chromosome. Chromosomes are divided into 4 types on the basis of the position of centromere. Number 1, metacentric chromosome. When the position of centromere is at the middle of the chromosome, is said to be metacentric. Thus, metacentric chromosomes have 2 equal arms. During
Anaphase.
this type of chromosome appears like the English letter V. Then submetacentric chromosome. When the location of centromere is slightly away from the middle point, so the 2 arms become unequal, is said to be submetacentric chromosome. Such chromosome appears L-shaped in anaphase stage. Then acrocentric chromosome, when the location of centromere is away from the middle point of chromosome, the chromosome is said to be acrocentric. Centric chromosome appears J-shaped in anaphase stage. Then telocentric chromosome. When the location of centromere is located at the end of the chromosome is said to be telocentric chromosome. A telocentric chromosome has 1 arm and it appears I-shaped in anaphase stage. Classification of chromosome on the basis of function. According to their function, chromosomes are 2 types, autosome and sex chromosome. Those chromosomes bearing genes for morphological features are known as autosome. In man, there are 22 pair of autosome. Then sex chromosome. Sex chromosome determine the sex of the organism. In man, 1 pair of sex chromosomes are present. Double XX chromosome indicate female, whereas males have XY chromosomes. Generally, Y chromosomes do not bear any gene. On the basis of staining ability, chromosomes may be of 2 types, euchromatin and heterochromatin. Euchromatin, the portion of chromosome become lightly colored when stained with basic dye, is known as euchromatin. Centromere, secondary constriction, is made of chromatin. Then heterochromatin, the portion of the chromosome which becomes dark stained with basic dyes, that is carmine or hematoxylin, is known as heterochromatin. Then role of chromosome in cell division. The principal substances of cell division is chromosome. Chromosomes are essential for the process of cell division and they are responsible for the replication, division and creation of daughter cells that contain correct sequence of DNA and proteins. Proteins make up one of the most important components of the human body. They are responsible for building muscles and tissues, growth and repair, as well as the synthesis of thousands of enzymes like DNA replication enzymes produced by the body. Protein synthesis steps and their successful completion is the responsibility of genes and are contained in chromosomes. During cell division stage, the chromosome is responsible for the replication and distribution of DNA amongst new cells. During mitosis, one cell divides into 2 daughter cells with equal number of chromosomes in each cell and move forwards opposite poles. If the chromosomes do not properly distribute, the cell division would not be effective. During cell division, gene is transmitted from parents to offspring through chromosome. chromosome plays a vital role in crossing over and mutation for evolution during cell division. It plays important role in determining male or female baby by sex chromosome. It plays important role in cell division, control, and structure formation by synthesizing different types of proteins and enzymes. During meiosis 1, the chromosome divide. As a result, the daughter cells get half the number of chromosomes from their mother cell. This cell is called gamete, which unites with another gamete to form zygote. Thus, diploid set of chromosome is maintained constant generation after generation.
Okay, your homework is classification of chromosome on the basis of the position of centromere.
I hope you properly understand our today's topics. You have to learn and memorize it properly. And if there is any problem, you must share with your subject teachers. They will solve. Thank you all to stay with us.